So fatigue in the skeletal muscle, it occurs due to the depletion of the ATP that is necessary for the muscle contraction. So when the ATP it becomes low, the muscle can not no longer generate energy for sustained contraction that leads to the muscle fatigue. So the accumulation of lactic acid is a factor in the muscle fatigue, but it is more commonly associated with the short duration, high intensity exercises rather than the low intensity exercise. So we know that the lactic acid it is build up of lactic acid buildup. It is a byproduct of anaerobic metabolism and it contributes to the muscle fatigue during intense activities. Moving to the next question, the red muscles it consists of. Well, the red muscles they consist of higher concentration of myoglobin. So these red muscles, they have high content of myoglobin and cytochrome with many mitochondria, rich blood supply, uh, and they are smaller in diameter. So the red muscle it consists of high concentration of myoglobin with oxidative fibers that gives it a red color. We know myoglobin it is a protein that stores the oxygen in the muscle cells and red muscles. Red muscles they are also known as slow twitch muscle. Red muscle fiber they are also known as muscle slow twitch fibers. So they contains more myoglobin to support prolonged contractions. So that is why they appear red in color due to the myoglobin content and increased blood supply. So they have a increased blood supply. That is why they appear red in color. They contain a higher number of mitochondria that are responsible for the aerobic energy production. Red muscles they have a few, whereas the red muscles they have a rich blood supply. Whereas the fast muscles they are also known, the white muscles they are also known as a fast twitch muscle fibers. So they are involved in activities like sprinting, weight lifting, etc. Coming to the next question, in smooth muscle fiber, the calcium binds to. Well, in the smooth muscle cells, the contractile process involves binding of the calcium to the calmoduli. So the answer is B. So for the first step in the smooth muscle contraction is increased level of the calcium. So there is increase in intracellular calcium level that can be triggered by various signals including transmitters, hormones or change in the membrane potential. So when the intracellular calcium concentration it rises, the calcium ions will bind to protein that is known as calmoduli. So this calmodulin it act as a calcium binding protein. So when calcium binds to it, it undergo a conformational change. So binding of calcium to calmodulin, it forms a complex that is known as calcium calmodulin complex. So this complex it is important for the smooth muscle. This process it is important for the smooth muscle contraction. So the calcium calmodulin complex it activates enzyme that is known as myosin light chain kinase. So this myosin light chain kinase it is responsible for phosphorylation. That is, it add a phosphate group to the myosin cross bridges within the smooth muscles. So once it get activated, uh, it allows myosin to bind to the actin filament and initiate the sliding filament mechanism leading to the muscle contraction. Coming to the next question, during BP measurement with sphygmanometer. So when measuring BP with sphygmanometer, it is important to ensure that the cuff and the device they are properly positioned to obtain accurate reading. So placing the cuff and sphygmanometer at the level of heart helps to ensure that measurement it is not affected by the gravitational differences between the heart and arm. So that can lead to inaccurate reading. So both cuff and sphygmanometer should be at the level of heart. Now the P wave in ECG recording it is caused due to the the P wave. So this P wave is caused due to the atrial depolarization. So P wave it would represents what? Atrial depolarization. That is the electrical activation of atria. So it corresponds to the initiation of the electric impulse at the SA node. So that spreads through the atria leading to atrial contraction. The other is QRS complex. The QRS complex uh, indicates the ventricular depolarization. So that is the electrical activation of the ventricles. That is the lower chamber of the heart. It includes Q wave, R wave, and S wave. So that leads to ventricular contraction. The other is the T wave that represents the ventricular repolarization. That is the recovery of the ventricular muscle cells following the depolarization. So P wave would represents the atrial depolarization. The SI unit of measurement of BP. Well, the SI unit of measurement of BP it's kilopascal. Well, the pressure in SI unit it is measured in pascals. BP in SI unit is measured in kilopascals. And 1 mm of mercury is equal to 0.133 kilopascal. That is the conversion. Hematocrit is defined as Hematocrit, it is defined as the percentage of blood volume that is occupied by the erythrocytes, that are the red blood cells. So it is important measurement of concentration of red blood cells in the blood, and it is typically expressed as percentage. And in normal male, the value of hematocrit it is around 45%. So hematocrit, it is a blood test that will measure the volume percentage of red blood cells in the whole blood. So it provides information about the proportion of the red blood cells in the blood. So a low hematocrit would suggest anemia that could be through nutritional deficiency, chronic diseases, bone marrow diseases, whereas the high hematocrit uh, may indicate polycythemia, that is condition that is caused by increased number of the RBCs, that can be due to the chronic lung diseases or certain smoking or certain bone marrow disorders. What is serum? Serum is plasma minus fibrinogen. So serum is the plasma minus fibrinogen and other clotting proteins. So when blood it is allowed to clot and clot retracts, it releases serum, which is initially the plasma without protein involved in blood clotting, that is without fibrinogen and other clotting factor. The venous return to herd during quiet standing, it is facilitated by all of following factors, except the answer would be D. The gravitational, we know the gravitational increase in arterial pressure does not enhance or improve the venous return during quiet standing. In fact, the gravitational it causes an increase in arterial pressure. It can be considered a potential hindrance to the venous return. So gravitational increase in arterial pressure, it would cause hindrance to the venous return. So when the person it is upright position, standing position, the gravity can increase the pressure within both the arteries and veins in the lower extremities. So this increased arterial pressure does not directly facilitate the venous return. So it can make it more challenging for the blood to flow from vein in the legs to back to the heart, as the blood have to work against gravity. So answer is D. Vitamin K dependent coagulation factors include. So remember this mnemonic. Uh, 2 plus 7 is 9. 2 plus 7 is 9 and not 10. So the mnemonic, the vitamin D dependent clotting factor are 2, 7, 9 and 10. Hope it is clear. All of following cells they are connective tissue region except except the Clara cells. So the Clara cells, not of uh, they are not of connective tissue origin. So these Clara cells, they are restricted mainly to the terminal and respiratory bronchioles. They are cuboidal, non-ciliated cells. Coming to the next question, the membrane protein clathrine. Well, the membrane protein clathrine, it is involved in receptor mediated endocytosis. We know clathrine, it is a protein that play a key role in receptor mediated endocytosis. So this process involves the selective uptake of specific molecules from the extracellular fluid by the cells. So the clathrine coated pits forms on the cell membrane, and these pits are coated with the clathrine protein. So the receptor mediated endocytosis, it is essential mechanism for cells to internalize specific substance like nutrients and signaling proteins by binding to receptors on the surface cell and then engulfing them in a vesicle coated with clathrine. The in a primary in the primary active transport, the covalent bonding with pi causes conformationally modulation of carrier protein. So primary active transport, it is a type of active transport in which the energy it is used directly to transport the molecules across the biological membrane. Typical against their concentration gradient. So the energy source for primary active transport, it is usually ATP. So which energy is used? ATP. So a common example is the sodium potassium ATPase. So sodium potassium pump is its example. So sodium potassium pump, it is a protein that is found in the cell membrane of most animal cells. So it actively transports 3 sodium out of the cells and 2 potassium inside cell for each ATP molecule used. So this pump, it is essential for maintaining the proper balance of sodium and potassium inside and outside the cell. So in the sodium potassium pump, the pump binds to intracellular sodium. The ATP, it is hydrolyzed to ADP, and the energy that is released during this process, it is used to change the shape of that of the pump, that is conformational modulation. So this change in shape that will allow the pump to release sodium outside the cell. So the pump then binds to extracellular potassium ions. So another conformational change is induced that allows the pump to release sodium, potassium inside the cell. So the covalent bonding with pi, it will cause conformational modulation of the carrier protein. In the secondary active transport, the movement of sodium is from higher concentration to low concentration. The secondary active transport, it is also known as co-transport. So there are two types of secondary active transport. One is symport. In symport, both molecules they move in same direction, whereas in antiport, the molecules they move in opposite direction. The example is sodium glucose co-transport. So that is responsible for the transport of glucose against its concentration gradient in the renal tubules of the kidney. So the example of secondary uh, glucose transport is the sodium glucose co-transport that is found in the renal tubules of kidney. So it is responsible for the reabsorption of glucose from urine back into the bloodstream. So in the extracellular fluid, there is high concentration of sodium due to the primary active transport by the sodium potassium pump. So SGLT will bind both the sodium and glucose on the luminal side of the renal tubules. So the binding of the sodium induce a conformational change, allowing both the sodium and glucose to be transported into the cell. So this movement of sodium from high concentration to low concentration gradient, it is driven by sodium potassium pump. So once inside the cell, the glucose it can be transported across the basolateral membrane in the bloodstream by a facilitated diffusion through the glucose transport. So the answer is A. The nerve conduction, it is the nerve conduction is an active self-propagating process that requires energy for the propagation of nerve impulses at constant amplitude and velocity. So the active conduction it is facilitated by the movements of ion, specifically the sodium and potassium across the cell membrane. Donan effect is due to, answer is D. We have already discussed this question back. The isovolumic relaxation phase of cardiac cycle it ends with the opening of the AV valve. So isovolume relaxation also known as isovolumetric ventricular relaxation. So it is a period in which both the aortic and pulmonary semilunar valves are closed. So during the isovolumetric uh, relaxation, the ventricles they are relaxing, and the pressure within them it is decreasing. So the AV valves that separates the atria from the ventricles they remain closed due to prevent in order to prevent the backflow of blood from ventricles to the atria. The semilunar valves that guards the exit from the ventricles into aorta, they are also closed. So the isovolumic relaxation period it ends with the opening of the AV valves. So when the pressure in the ventricle it drops below the pressure in atria, the AV valves they get open, allowing blood to flow from atria to the ventricles. So this marks the transition from the isovolumic relaxation to ventricular filling phase. during which the ventricles they are filled with blood from the atria. The QRS complex would indicate, already discussed, it would indicate ventricular depolarization. When a normal person changes position from standing to lie down, the following changes is seen. So when the normal person position changes from standing to lie down, the venous return to the uh, heart, it increases almost immediately. So in the standing position, the blood can pool in the veins of lower extremity due to the effect of gravity. So when the person lie down, this pooled is returned to the central circulation, increasing the venous return of the heart. So this venous return helps to maintain the cardiac output and blood pressure when changing position. During diastole, the arterial pressure it is maintained by during diastole, the phase of cardiac cycle when the heart is relaxed and it is filling with blood, the arterial pressure it is primarily maintained by elastic coil of the aorta. So during systole, the heart pumps blood into aorta and large vein, stretching their walls to accommodate the incoming blood. So when the systole ends and diastole begins, the heart relaxes. So during this relaxation, the elastic walls of aorta tends to returns to their original size. So this is known as elastic recoil. So as the wall of these elastic vessels will recoil, they will exert pressure on the blood inside, helping to maintain the continuous forward flow of blood and arterial pressure during the diastole. The best method to assess the adequacy of replacement of fluid in case of shock is increase in the urine output. So increase in urine output indicates that uh, kidneys they are receiving sufficient blood flow and are capable of filtering and exchange and excreting waste products and excess fluid. So it is a quantitative and relative reliable indicator of organ perfusion. So in case of shock, maintaining or increasing the urine output, it is positive sign that circulatory system it is responding to the fluid replacement. The urine output it is a practical and reliable method to monitor the response to the fluid replacement in case of shock, as it will represents the kidney overall function organ perfusion. A synthetic antidiuretic hormone that controls bleeding in dental patient with the type 1 bone Willebrand disease, it's the desmopressin. So it is a synthetic antidiuretic hormone that is control used to control the bleeding in patients with von Willebrand disease. So it helps to improve the platelet function and enhance the blood clotting. The cardiac muscle they have a the cardiac muscle they have a long refractory period. So that means that after the cardiac muscle fibers contracts, it takes long time before it can contract again. So during the refractory period, the muscle fiber cannot respond to another stimuli no matter how strong it is. So a long refractory period of cardiac muscle it is essential for the proper functioning of heart and its ability to pump blood effectively. SA node is known as the pacemaker of heart because of fact it generates impulses at higher rate. SA node is also known as pacemaker of heart because it possesses the ability to generate electrical impulses at highest rate. So remember the SA node, it generate electric impulses at the fastest rate among all the components of the heart electrical conducting system. So SA node, it is the natural pacemaker of the heart because it is capable of generating impulses spontaneously, and it does so at highest rate, setting the pace for the heart rhythm. The increased venous return to heart, it will cause uh, increase in end diastolic volume. So end diastolic volume, it is the amount of blood in the ventricles of heart at the end of filling phase, just before they contracted during systole. So imagine a person who has been resting for a while. In this state, the body oxygen demand is relatively low, and veins, they are returning blood to heart at a steady state. So this person and diastolic volume is considered baseline. Now, what happens during exercise? Increase venous return. When person starts to exercise, the muscles are working harder and require more oxygen. So this increased muscle activity that results to increase in return of the deoxygenated blood to the right atrium. So the increased venous return means that there is more blood filling the right atrium during systole. As a result, the right ventricles receive a large volume of blood during its filling phase. So this increased blood volume in the ventricles at the end of diastole, it is reflected as end diastolic volume. So the heart responds to increased diastolic volume by increasing the force of contraction during the systole. So that allows the ventricles to pump out larger volume of blood, that is known as stroke volume. So increased venous return as during exercise, it leads to increase in the end diastolic volume. That in turn contributes to increased stroke volume and the cardiac output. The arch of aorta, it begins at the level of sternal angle, that is also known as angle of Lewis. The right ventricles, they receive blood from the right ventricle, they receive blood from the superior vena cava. So superior vena cava, it's a large vein that carries deoxygenated blood from the upper half of body back to the heart. So blood from the superior vena cava flows into right atrium. From the right atrium, the blood then passes from the tricuspid valves into the right ventricle. So the right ventricle, it is responsible for pumping this deoxygenated blood into the pulmonary artery that carries it to lungs for oxygenation. The membrane protein clathrin, it is involved in, answer is B. Already discussed. The majority of the blood sodium, it is present in the extracellular fluid. So sodium is the most abundant cation that is present in the extracellular fluid. QRS complex indicates the ventricle depolarization. Already discussed. The isovolumic relaxation phase of cardiac cycle, it ends with the opening of the AV valve. Already discussed. Which of following is not a sarcolemmal protein? The answer is D. Sarcolemmal proteins, they are proteins that are found in sarcolemma, that is the cell membrane of the muscle cells. So these proteins, they play a role in muscle structure, functioning and communication. Lectin, it is a large multi-domain proteoglycan that is found in extracellular matrix. It is not a part of sarcolemma or the sarcolemmal protein. In human, the least used physiological response to low environment temperature is the pilorectin. So pilorectin refers to phenomenon where the small muscles at the base of hair follicles they contract, causing hair to stand on the end. So this is also known as goosebumps. It is the physiological response to cold or emotional stimuli. So that occurs due to the sympathetic nerve activation. So it is commonly it it does not provide it is not very useful in the humans. So the least useful response to low environment temperature is the piloerection. Other options like shivering, it is a mechanism by which the body generate heat by rapidly contracting and relaxing the skeletal muscle that helps to increase the body temperature. Vasoconstriction, that refers to the narrowing of blood vessels in response to cold, which helps to reduce the heat loss by decreasing blood flow to the skin surface. And thyroxine, it plays a role in regulating the body's metabolic rate and can be increased in response to cold to generate more heat. The heme, it is converted into bilirubin mainly in the spleen. The breakdown of heme to bilirubin, it occurs in the macrophages of the reticuloendothelial system that occurs mainly in the spleen as well as in liver. So answer is spleen. The blood supply of SA node is right coronal artery. The cell junction that allows exchange of cytoplasmic molecules between two cells, they are the gap junctions. Already discussed, this question was already discussed before too. The distribution of blood flow is mainly regulated by arterioles. Arterioles, they are the major site of resistance to blood flow in the circulatory system. So by changing their diameter, arterioles can significantly influence the resistance to the blood flow in the different vascular beds. So that determines how much blood is delivered to various tissues. So option B is correct. Which of following can be determined by calculating the clearance of para-aminohippuric acid? The answer is effective renal plasma flow. So para-aminohippuric acid, it is commonly used method to estimate the effective renal plasma flow. So the rate at which para-aminohippuric acid is excreted in urine, it reflects the rate of plasma flow to the kidneys. So this is an important ERPS. It is important parameter for evaluating the kidneys function and it is used in the determination of renal blood flow and glomerular filtration rate. Which of following statement is true about the troponin C? Troponin C, the is one of the main component of the troponin complex. So it binds calcium ion, and in presence of calcium, it initiates the muscle contraction by permitting the interaction of actin and myosin. So when the calcium it will binds to the troponin C, it will cause a conformational change in the tropomyosin complex, allowing the myosin to bind to actin and starts the contractile process. So it permits the interaction of actin and myosin.